So let me get started. Welcome. This is our June 8th meeting of the Multiscale Modeling and Viral Pandemics Working Group. It's been a while since we last had a meeting. I appreciate people coming back. It's very kind of you. And today we have a single talk from the University of Virginia, which we'll get to in a minute. As always, Tom Helikar and I are available to take questions, comments, suggestions. Jim Sluka and Lorenzo are also available to handle situations. I think Lorenzo is away this week. I think he's on holiday, so he may not be here today. We have our various communication channels. People have seen this slide before. They work better when you use them. We should probably add the GLIMPRINT website to this, right? Tom, that would be good. And I would appreciate your help trying to build a little bit of an audience for our speakers. We have that MSM viral tag to use in annotation and Twitter. Maybe we can use that to build a little bit of recognition. I'll be seeing quite a few of you in a few weeks in Bethesda, June 28th to June 29th. I hope that we can highlight some of the things you've done here at that meeting, maybe build some support for moving forward with that. We still have at Indiana University two primarily senior positions in medical digital twins. If people are interested, please let me know. Those hires could be at the associate professor or full professor level. If you're interested, we definitely want to talk to you. I also will say that we still have openings in our CompuCell workshop for the summer. We are doing this entirely virtual. We've expanded the time frame to 2 weeks to try to make it more accessible to people, so it's not so much of a fire hose. If you are a CompuCell user and you're willing to help us be a teaching assistant or help us with a hackathon for a day, we'd be grateful. If you want to take it or you want your students to take it, we'd be grateful too. We have 150 people registered so far, so it'll be interesting to see how that works. But we definitely are open to more people. Tom has been working hard developing GLIMPRINT, Global Alliance for Immune Prediction and Intervention. We have the website, we have other things. Anybody who's willing to give some time and energy to building GLIMPRINT would be much appreciated. Tom has been doing a lot of lifting, and it would be super helpful to have support for that. We will have a steering committee meeting tomorrow at 11 a.m. Focusing on GLIMPRINT. The question will be, what do you personally want to get out of GLIMPRINT? What do you think GLIMPRINT could do for you? Which is not quite the same question. And potentially what GLIMPRINT could do for the community. We'd like to be able to build a little bit of an action plan. Since there was a community decision to build this institution, we now need to start thinking about what it will do and how to deliver on that. And so we'd appreciate help for that. Are there any other announcements before we go forward to the talk today? No. Okay. Coming up for meetings on June 22nd, we have Harm from the National Institute of Public Health and the Environment in the Netherlands, who's going to be talking about modeling toxicant-induced defects. They have a big project. Lisbeth Garris and the VPH people are very heavily involved with that. So that'll be pretty interesting. And then July 6th, we have Tom Yanklov talking about his amazing coupled clinical, experimental, and computational work, essentially building personalized models of breast cancer therapy and response from Texas. And Reinhard, you are tentatively put down for July 20th to tell us about what happened in Paris, you and Anna. That could be updated, but that's what we have.
Yes, I can do that.
Great, that will be wonderful. Okay, So I don't want to take any more time away from our speaker. We're going to be hearing about muscle regeneration agent-based modeling, which have, of course, a lot of overlap with immune system and the idea of virtual tissues and potentially then using those virtual tissues for personalization of therapy. And so I will turn it over to you.
So as Dr. Glazer mentioned, my name is Megan Haase. I'm a PhD candidate at the University of Virginia in the Multiscale Muscle Mechanophysiology Lab. And today I'm excited to talk to you about my agent-based model of muscle regeneration that focuses on predicting how cytokine dynamics can alter muscle regeneration outcomes. So to jump in, muscle regeneration is really essential for everyday life. So if we think about everyday use, typical exercise, we get these little micro tears that induce acute muscle damage. But skeletal muscle has this really remarkable regenerative capability to repair itself. But with chronic micro tears or in certain disease states, we can see more extensive muscle injuries, such as muscle tears and muscle strains. And these types of injuries are one of the most common complaints to orthopedics. And because muscle regeneration is a pretty highly coordinated cellular process that depends on a variety of different signaling factors, developing treatments for muscle injury can be fairly complex. So to dive a little bit into this whole regeneration cascade, if we start by looking at a muscle fascicle, we can see these healthy muscle fibers which sit within the extracellular matrix. And upon some initial muscle damage, we have this inflammatory phase where neutrophils are moving in to respond to the damaged fibers. And then we'll also see pro-inflammatory macrophages, followed by fibroblasts that will move in to help secrete collagen to repair the damaged ECM, as well as these satellite stem cells that help repair the fibers. They'll start to become activated. And then we'll also have the remodeling phase where some of these pro-inflammatory macrophages will transition into an anti-inflammatory state, and we'll have differentiation of satellite stem cells. And then assuming we have all of the appropriate signaling factors and microenvironmental conditions, these satellite stem cells are able to fuse to fibers and other differentiated satellite stem cells to restore the muscle. And so this is a fairly complex process, so we need good tools to really understand all of the aspects of this regeneration cascade. And so what our lab does is we implement computational modeling to study these complex dynamics as well as gain new insights into muscle regeneration. And to do this, we start by leveraging literature from a lot of great publications. And using this literature, we're able to define physiologically based rules for each cell type. And so once we put all of these different cellular behaviors together to define all of our agents within this agent-based model, we're able to gain new insights into different cell and cytokine crosstalks, as well as look at testing alterations in different signaling and cytokine dynamics, as well as predict time-varying information, specifically spatial information that is a lot more difficult to get experimentally alone, especially at a variety of different time points. And through the use of all of this, we're able to generate new hypotheses for in vivo experiments, as well as hypothesize targets for different types of muscle regeneration treatments. And so if we think about applying this cascade to an agent-based model, so some example model agents, if we think back to that satellite stem cell, so upon damage, there's a certain amount of HGF that gets secreted by these damaged fibers, and then that signals for the satellite stem cell to undergo activation. Similarly, there's a different subset of cytokines within that microenvironment that can then signal cell division, as well as cytokines that can signal differentiation. And once these differentiated fibers are nearby damaged fibers or other differentiated satellite stem cells, they're able to fuse to repair. Also within the model, we can have neutrophils, and those neutrophils move towards areas of necrosis and then secrete MMP to break down these areas of damage as well as portions of the ECM, and then they can undergo cell death. And then we can also see the macrophages, they chemotax along an MCP gradient towards areas of necrosis, as well as can help phagocytose apoptotic cells. And so because of the power of agent-based modeling, our lab has also developed prior models that have studied muscle regeneration. The first of which used the NetLogo agent-based modeling platform to kind of build this preliminary model of muscle regeneration. But this model had pretty limited diffusion as well as simplified cell dynamics, which made it difficult to fully grasp the crosstalk between a lot of the signaling factors and how the cells were spatially interacting. Our lab has also developed a model of Duchenne muscular dystrophy using a Java-based platform for agent-based modeling, as well as looked at studying muscle regeneration in the context of volumetric muscle loss, where a portion of the muscle fibers are removed following a traumatic injury. And these models, while they were insightful, they did not include spatial diffusion of cytokines or spatial dynamics of inflammatory cells, which limited our ability to fully understand those dynamic interactions. And so because of this, it really motivated me to want to build an agent-based model that really focused on spatial cell dynamics and cytokine diffusion and how we could leverage alterations in these cytokines to potentially enhance regeneration outcomes. So the goal of this project was to build an agent-based model to predict regeneration outcomes with altered cytokine dynamics. And to do this, I first needed to develop a computational model of muscle regeneration that includes cellular and cytokine spatial dynamics, as well as the microvascular environment. I also needed to calibrate the model to capture cell behaviors from published experimental studies, as well as validate these model outcomes, and then conduct in-silico experiments to predict how altering cytokine dynamics could impact muscle regeneration. So this is kind of an outline of what I'm going to talk through today, and so I'm going to go ahead and jump into the development of the computational model. So all of the complex cell behaviors that we talked about in the background, as well as a variety of different cytokines, were modeled spatially. So this included the inflammatory cells, such as neutrophils, monocytes, and pro and anti-inflammatory macrophages, as well as the satellite stem cell lineage, starting with quiescent satellite stem cells that could become activated, differentiated, and then fuse. Fibroblasts, as well as myofibroblasts, both of which could secrete collagen within the microenvironment. And then the microstructures of the agent-based model include the extracellular matrix, which is made up of collagen bundles, and each has an associated collagen density, the muscle fibers, as well as the capillaries, and then an array of different cytokines, each of which were included as necessary to guide agent behaviors. And so some example behaviors within the model include cell proliferation and differentiation, quiescence, apoptosis, migration, and activation, as well as secretion and uptake of cytokines as different behaviors are performed by the cell to simulate the binding interactions with these signaling factors. So this kind of shows a general high-level overview of how the model sequence is performed. So we start with initialization, followed by different cell recruitments, and then each cell behaviors, and then the resulting microstructure changes. And so for initialization, we start by importing muscle histology. So this is showing a cross-section of muscle, which is H&E stained. We pick a subset of that, and then we import it into MATLAB to differentiate between muscle fibers and ECM. And then this gets imported into CompuCell 3D, where we're then able to assign different microstructure elements. So for example, the muscle fibers are the dark pink, the ECM is shown in white. We also have capillaries in blue, and then a lymphatic vessel, which is draining the cells and cytokines within the microenvironment nearby. And then following this, the user is able to define a preset amount of damage. And this is based on the extent of the injury that they want to model, as well as the type of injury, which dictates how the injury occurs within the model. So, for example, for cardiotoxin-induced injury, which is pretty common within the literature, there's an area of necrosis and then a radius around that, which is simulating the cardiotoxin injury diffusing out. But other options could be a freeze-induced injury, where it would take a subset where the freeze probe touched the muscle, and it would damage all of those fibers, as well as result in apoptosis of any cells within that damaged area. And so the necrosis is represented by this light pink fiber. And then following this, we also have a randomized placement of baseline cells, such as quiescent satellite stem cells, resident macrophages, and resident fibroblasts. And then upon start of the simulation, these damaged fibers secrete damage factors such as HGF and TGF. And so another new aspect of the model is we included microvascular growth and remodeling because of the key role that it plays in muscle regeneration. So this image is showing fibers in red and then capillaries adjacent to these fibers. And so this was incorporated into the model. And so the areas nearby necrosis or fiber damage, any capillaries that were adjacent to damage would become fragmented, meaning that they were not able to perfuse any cells or cytokines into the microenvironment. But with sufficient cytokines at the location of the capillary, such as VEGF, they could undergo angiogenesis throughout the simulation so that they are able to continue bringing in nutrients and cells to the microenvironment. We also include the formation of new capillary formation. So if there is sufficient cytokines to signal sprouting capillaries, that gets included in the model, and then those are able to secrete cytokines as well as bring in cells to the microenvironment. And then another piece for accurately modeling the dynamics of these cytokines was to have more dynamic ECM properties. So as fibroblasts are moving throughout the environment, they are secreting collagen, which is altering the collagen density of each ECM element. And so as the collagen is changing, this is also altering the diffusivity of the cytokines within the microenvironment. And so to account for this, we use the formula shown here, which includes the cytokine and fiber radius, as well as the fiber volume fraction and the cytokine diffusivity in free solution, so that with each Monte Carlo step, we can calculate the new diffusivity of the cytokine in the matrix to more accurately simulate their dynamics. And so kind of putting that all together, you can see that it becomes a fairly complex flowchart of agent rules that occur. And so because we don't have time to necessarily step through all of this, I wanted to go through an example of one of the agents. So if we think about these satellite stem cells that we've kind of talked a lot about because of the crucialness of this cell within muscle regeneration. So what it does is within each step of the model, it checks if that satellite stem cell is active. If it's not, if there's enough activation signal for it to become activated. If so, that satellite stem cell is inactive and it removes the amount of cytokine at that location of the cell that induced activation. Similarly, it's checking for enough damage signal, if it should become quiescent, if it's near a damaged fiber, if it can fuse, if there's enough collagen at that location to kind of hold in the structure of that fiber. It also checks for division, and if it can divide, it can do so symmetrically or asymmetrically, as well as if it has enough signal to differentiate or if there's signal to undergo cell apoptosis. So looking at how this all kind of comes together, so if we look at the cross-section, you can see the necrosis, it gets cleared, and then over time gets regenerated. And then we can also see how the cells are behaving throughout this. So at the beginning, we will get an influx of neutrophils followed by macrophages, and then satellite stem cell and fibroblast proliferation, as well as differentiation and fusion. We can also look at how the damage signal is changing over time as cells secrete it, as well as it diffuses and decays over time. And then we can also look at the interplay with all of the other cytokines and how each of them has their own temporally based dynamics, and how as the cells are changing, it's also altering these cytokine dynamics, each of which has their own unique defined decay and diffusion parameters. So once the model was developed, the next step was then to calibrate our model to experimental studies published in the literature. And so we chose to use a fairly new calibration tool called CaliPro, which is specifically designed to calibrate complex stochastic biological models. And so this is kind of a high level overview of the protocol, and then I'll dive a little bit further into some examples for my model. So the general protocol starts with user inputs as well as experimental data used for calibration, the model, and then the unknown parameters. And then you do some type of sampling over your unknown parameters, such as Latin hypercube sampling, where you generate a general range for each of the unknown parameters and then get unique parameter sets to run model simulations. You then evaluate these model simulations against criteria set using the experimental data. So for example, setting a criteria to fall within experimental standard deviations. And then you redefine the parameter space using either a high density region or alternative density subtraction to then limit the parameter space for the next iteration. And then this continues until you're satisfied with some termination criteria for calibration. So if we think back to all of the complex satellite stem cell rules, we can see that there are a variety of different aspects of the rules that are fairly difficult to quantify experimentally. So like how much of a signal does it take to undergo division or differentiation? So these are just a couple of examples of how uncertainty played a role in what needed to be calibrated. And so we use parameter density estimation as well as partial rank correlation coefficient to calibrate. So we had 52 unknown parameters. We then used Latin hypercube sampling to generate 600 unique parameter sets, and each of which was run in triplicate on the cluster. And so the top plot is showing all of the different simulations that were run. And then the dots are representing the experimental data, with the middle one being the average and upper and lower representing the standard deviations. And so then if we set our criteria to be that we want it to be nearly within the standard deviation from experimental data, you can see in the lower plot how it narrows in on those parameter sets that achieve that criteria. And then from there, we're able to look at all of the different unknown parameters and see the density of the passing runs as well as the density of the failing runs. And then using alternative density subtraction, we subtract these two densities, which then gives us limits for the next Latin hypercube sampling to generate new parameter sets to test. And then in addition to this, we also use PRCC to look at how the sensitivity of all of our unknown parameters was impacting certain outputs, such as how the cross-sectional area of the muscle tissue was recovering. And then this helped us better understand the real meaning of these parameters and how they may have impacts and interplay between different parameters that could potentially be problematic as we were trying to fine-tune certain parameters. So following this iterative process, we were able to calibrate our muscle cross-sectional area recovery, as well as the satellite stem cell and fibroblast cell count for nearly within all of the standard deviations from experimental results. And so once this was calibrated, the next step was to take separate outputs that we didn't include in the calibration scheme and see if they were still able to align with experimental results. And so we looked at total macrophage count, which was not included in the calibrate iteration, and we found that our total macrophage cell count fit within the experimental standard deviations. As well as looking at M1 and M2 count, we found that these results followed experimental trends. And then neutrophil and capillary per area, similarly, without being in the calibration scheme, were still validated against separate experimental results. And so this gave us confidence that our model was able to capture these complex cell dynamics, but the next step was to start altering model inputs to see how we could capture other experimental conditions. And so this figure is showing a variety of different experimental conditions that we simulated with the model. And so some of these were altering different cell dynamics, such as if we deplete neutrophils or macrophages, or if we start knocking out different cytokines, or if we prevent angiogenesis from occurring, or if we start to inject different cytokines. And so as you can see here, the majority of our simulations aligned with the experimental studies. So with the literature studies shown on the top triangle of the figure, and then the model ones are at the bottom triangle. And so this was exciting to see that we could start to alter model input conditions and still see alignment with experimental results. So then we wanted to dive a little bit deeper into some of these simulations, because now we had the opportunity to not only compare the outputs from experimental results, but look at new outputs that the model is predicting that weren't found experimentally, as well as look at additional time points that weren't captured with the experiments that were published. And so the first example of this is we use the model to dive a little bit deeper into how preventing angiogenesis altered muscle regeneration, as well as how there was a response to different VEGF injections. So the first plot is showing we simulated a variety of different VEGF concentrations at the 5 days post-injury. And so you can see the peak in VEGF concentration that has a sustained increase over time. And we found that there was a slight dose dependency with VEGF up until a high injection, where we found that increasing VEGF injection no longer was beneficial to cross-sectional area recovery. And then looking at this also helped us better understand how injecting VEGF at different levels would alter capillary counts, as well as different cell dynamics, such as macrophage cell count, satellite stem cell, and fibroblast cell count, specifically how this alters peak cell counts and timing of these peaks. And then in addition to this, we can look at with hindered angiogenesis, and if we think about injuries where you wouldn't get good capillary formation, how that could alter muscle regeneration dynamics, particularly how that's altering cytokine dynamics such as HGF and MCP, which can provide insight into how we may want to address this with different types of treatments for these injuries. The next thing that we looked at was what would happen if we knock out different cytokines within the model. And so what we did here was we knocked out individual cytokines, and then we looked at the response in all of the other cytokines at a variety of different time points. So for this, we knocked out monocyte chemoattractant protein and compared it to the control simulation. And then we found that not only did this cause alterations in all of the different cytokines, but each cytokine had its own unique response that varied throughout time. And we found that this was consistent with all of the knockout conditions that we did. So we also looked at TNF knockout and IL-10 knockout. And it was interesting just to see how some would be lower at like 12-hour time points, but then by the 28-day time point, they were significantly elevated. And so what this was telling us was that these cytokine dynamics, there's a lot of crosstalk that goes into each one of them, and that better understanding alterations in individual cytokines could be useful for understanding how we could treat muscle injury. So then this guided us to want to better define these relationships. And so what we did here was we used Latin hypercube sampling and pressure rate correlation coefficient, which we had started with calibration, but then we wanted to apply it for something different. So we use that to look at relationships between cytokine parameters and key regeneration metrics. And so we took all of our cytokine decay and diffusion parameters, and we sampled them using Latin hypercube sampling, and then ran different simulations, and then used PRCC to see what the sensitivity of those cytokine parameters would be, and then what alterations would result in better cross-sectional area recovery, different cell dynamics, such as how the satellite stem cells would differentiate, as well as how the inflammatory cells would respond. And so following this, we then had a better understanding of links between cytokine dynamics and cell outputs, and how those varied throughout time. And so using this, we then generated hypotheses for different cytokine alterations that could potentially enhance muscle regeneration outcomes, such as cross-sectional area recovery and satellite stem cell count. And so we then applied this. So this is showing all the different perturbations that we ran. So we had our control simulation, and then we tested lower HGF decay, higher TGF, MMP, and MCP decay, as well as higher MCP diffusion, lower VEGF decay, and then a combination of cytokine alterations. And so what we found here was that all except for one of the cytokine alterations resulted in enhanced cross-sectional area recovery compared to the control case. And then this also allowed us to look at how each of these cytokine alterations was impacting cell counts, such as M1 and M2 response, and fibroblasts and satellite stem cell count, as well as capillaries. And so we could see with individual cytokine alterations that we had changes in fibroblast peak as well as satellite stem cell peak and where that peak was occurring. And then the really interesting thing was that when we took these perturbations and combined it into a cytokine combination alteration, that we saw the most enhancement in cross-sectional area recovery, as well as an increase in fibroblast and satellite stem cell proliferation. And so this is really showing us how we can leverage this model to develop new types of therapeutics and try and optimize treatments for muscle regeneration. So to kind of conclude, we used Latin hypercube sampling and partial rank correlation coefficient, but to calibrate the model as well as to help identify potential therapeutic mechanisms. We found that cytokine dynamics play a really essential role on muscle regeneration, with any single knockout condition leading to cascading impacts. We also found that this agent-based model allowed for deeper exploration of temporal effects of different cytokines on regeneration metrics. And then our in-silico perturbations found that combining cytokine alterations, specifically looking at how we can alter decay and diffusion parameters, could potentially enhance regeneration outcomes. And then lastly, future work, we plan to explore different treatment optimization methods as well as alterations with various injury types that are used in the literature. And with that, I would like to thank my lab members, as well as my CompuCell 3D collaborators, my undergraduate researchers, and my funding sources. Thank you. Any questions?
Okay, great for the very compact presentation of a very complex model. Questions? Floor is open for questions. If somebody has trouble unmuting, let me know. I should have given people permission to unmute, but in case you can't, please ask.
Is there any consideration of learning from or comparing to wound healing modeling?
Yeah, that's a great question. Right now, the agent-based model doesn't have like any form of like reinforcement, like learning or machine learning aspects for the agents. That is something that we've like considered adding, but at the current state of the model, it doesn't necessarily take that into account.
I'm looking specifically at mobilization and migration that are common to both.
Okay, interesting. Like cell migration over.
Yes, exactly.
Yeah, because that's definitely a key part of the model is how these cells are migrating along different like cytokine gradients. And so right now those are kind of just like set rules that, you know, certain cytokines will just migrate along their respective gradient to like higher levels of concentration.
You started with muscle injury during exercise, and I would call it part of sports injury, which then calls for looking for wound healing.
Right. Yeah, I mean, muscle regeneration is simulating like a form of wound healing. Yeah, in a sense, but it's like specifically for like how these muscle cells are regenerating.
Yeah, clear. Thank you.
Yeah, thank you.
In your model, is the ECM basically fixed? And does that force the fibers to reform with the same basic pattern they had at the beginning?
Yeah, that's a great question because it's actually something that we're working on right now. So this iteration of the model, the ECM is fixed. So it does force, like if you saw in the simulation, like the fibers will only form where they previously were. So this next iteration of the model that we're currently working on is kind of also including aspects of muscle hypertrophy. So making the ECM more dynamic and the fibers more dynamic in the sense that once the damage is cleared, it can then fuse wherever. But that, because it creates a lot more rules have to be incorporated to include that type of dynamic, because then you have to think about like, oh, well, the fibers then might start to like merge together when like that doesn't happen physiologically, as well as like dictating where fibers can fuse and things like that. But yeah, no, that's a great point, and that's something that we're currently adding in.
And this is a 2D model.
Yes.
What do you do at diffusion on the boundaries? Do you set zero concentration at the boundary, or are you wrapping diffusion around you periodic boundaries or something?
Yeah, we currently have periodic boundaries.
Right. So it's mimicking an infinite sheet.
Right.
Something
It would be cool to someday have this as a 3D model though. That is like, it would be beneficial just to more accurately simulate some of the fibers.
As soon as you do that, your Latin cube sampling is going to be a real problem.
Yep. Yeah.
Simulations are going to be 1,000 times slower.
Yeah.
In the Latin hypercube sampling, how do you avoid missing local minima, or even missing a global minimum, instead iterating down onto a local minimum?
I'm not 100% like certain on how we avoid that. We're kind of just looking to like find a solution. And yeah, go with that. Yeah.
Okay. Other key questions here. And I was thinking you're trying to do this in 3D is very attractive in many ways, but the system is very anisotropic. And so you're taking this sagittal section. When you extend in the perpendicular direction, then you're extending along the fibers. And so then you have to think about what that does. It'd be quite interesting to do that. I had a sort of a basic question, which is, it's not that these models aren't complex enough and informative enough, and maybe it's not relevant, but you use the same initial condition, right? You have one image from one sample that you've been using to generate your initial conditions. Do you think that trying different samples which have slightly different geometry or larger samples would make a difference, or do you think this is big enough and typical enough that it's sufficient to represent the variety of muscle that you're likely to encounter?
Yeah, that's a great question, and also something that we've started working on. So as I've begun taking more histology images, we're importing different images just to see how much variation there is, as well as if we start like looking at different capillary to myofiber ratios, like what is that doing to model simulations? We found that if we change the histology image, it's still able to regenerate, which was good for us because that means that our calibration wasn't dependent on the one histology image. But yeah, we do like to change it out to see different results with different initial conditions.
I mean, if you compare histology images, I mean, looks here like the domains are pretty much similar size. The distribution is pretty similar. In the case that I would have been more familiar with, for example, like retinal vasculature, when you do fundus images of the retina, they differ enormously between individuals. I mean, really a lot. So much that you wouldn't expect that if one of those networks worked, the other ones would work. There's really a huge variation. Maybe here in this case of muscle, individual to individual, there isn't so much difference. And yeah. So I just don't know if you look at, this is more, it's not a modeling question, it's a histology question, which is if you took 10 patients, maybe different in age, difference in obesity, how different would the typical size of the myofibers be and their distribution?
Yeah, for the most part, if you're looking at healthy muscle, all from like the same, like from mice from the same muscle, even sometimes like if we look at like TA muscle versus like gastroc, the muscles have a similar look, a similar fiber area overall. If we started looking at like injured muscle tissue or like disease state, then we would start seeing like more non-uniform muscle fibers, more extracellular matrix in between the fibers, things like that. A lot of the fibers would be smaller. But generally with healthy muscle tissue, they're pretty consistent in terms of fiber distribution.
I mean, I guess in mice, they don't live very long. And so some of the age-related things that you get in humans might not show up, although I guess mice age a lot if you keep them going for a while. I don't know how far you can keep them going, whether there's an aged muscle model in rodents.
Yeah, and that's something like we haven't necessarily incorporated some of the like aging muscle aspects, but definitely for sure they would there are age-related effects to muscle regeneration as well as just how muscle changes with age. Yeah.
Other questions? Comments? Let me look at that. Jim, go ahead.
A kind of a quick practical one. How long does one of these simulations take to run?
On the cluster, they take between 2 to 3 hours.
A single thread or multi-thread? You know how many CPUs you're using per simulation?
I don't. I know usually we'll like request, I think like 40 cores and then have like all of the simulations running parallel. So then
it's a pretty big calculation. Yeah, I can imagine it takes a while with all those diffusing fields and stuff. It's not going to be a fast process, and that's why a big reason for not trying to go to 3D. Did you? So there's certainly variation across other muscle fibers, and as James was asking about, is this a typical layout? Did you look at effects of changing where you set the initial damage across this particular initial field?
Yeah, so each simulation, it's a randomized placement of damage. So every time it's different. We did also, one of the things that we've been kind of exploring a little bit more is location with respect to like the lymphatic vessel, because that's like clearing a lot of the cells and cytokines. So like if we damage near the lymphatic vessel versus farthest away. Similarly, like if we do it like closer to more capillaries, or if we increase that ratio and things like that, to see how much of like the microvascular structure is impacting the muscle regeneration outcomes.
Have you looked at running the same damage twice to see how reproducible the damage is, or the recovery from damage? Does it seem to do the same thing every time?
Just because of some of like the stochasticity of the rules, there is a little variation, but generally we'll see within like 5 to 8% recovery like variation just from like stochasticity. But that was a kind of when we were going through like the calibration, why we did like 3 replicates each, just to see like if there was something stochastic that happened with the simulation, that we could kind of see if it was consistent across the other simulations as well.
Other questions? Sharon, go ahead.
Yeah, just a question about your progression through these different simulation platforms. It looks like you've had a big trajectory. You started with one, got what you could out of it until you ran into its limitations, and then tried another and another. I think this is number 3. I wonder if you have any more to say about what that process was like, and also where do you think CC3D stands? Like, are you running into limitations there? Do you need to do something even better, or is this it? Is this pretty much for now giving you everything you need?
Yeah, no, I played with my fair share of agent-based modeling platforms, for sure. So the previous models were kind of like before me in the lab. So I did some work. I played around like with NetLogo when I was first introduced to agent-based modeling. And it's definitely a cool tool for like learning the process of agent-based modeling, but the complexity is fairly like limited. So I moved away from that, tried Repass Symphony, trying to work off of a previous model from the lab. And that was kind of where I ran into the issues of like, I was trying to ask a question that was pretty far removed from what they were trying to ask. And while the model was built perfectly for what they were exploring, for my research question, it was no longer, really applicable. And Repass Symphony, in terms of like some of the diffusion dynamics, I was running into roadblocks, and that's why I was starting to look for other platforms. In CompuCell 3D, I went to the workshop, so this image is actually, the first one is like from the CompuCell 3D workshop, and looking at it now, it looks very funny because it's it progressed so much more. But it was very easy to learn, and there were so many resources to help with building the model, as well as even as I've continued to work on it, there's so many people that I've been able to reach out to get further guidance. So I'm planning on sticking with CompuCell 3D because even if I run into roadblocks, there are people to help me overcome those roadblocks. So I'm pretty happy with the platform.
And you think those roadblocks are just more like figuring things out, not actual limitations?
Yeah, not really limitations. There's typically a way around it.
Cool, thank you.
Yeah.
Hayden, did you have a question?
Not particularly a question. I just thought that this was a very good talk for illustrating the overall modeling workflow as well, from identifying a problem to deciding which software you're going to use. That's actually something discussion that gets skipped a lot in just talks like this. So it was nice to see some discussion of a couple of attempts of finding the one that was right. And we're obviously glad to hear that CompuCell was the silver bullet in this case, but no, so Megan, I think I'm going to follow up with you after the talk, if that's okay. I'm organizing the workshop for this year. We may be interested in having you do a guest presentation during the second week if you're available and interested in participating, but we can talk about that offline after this.
Yeah, sounds great. Thanks.
I mean, maybe you could talk a little bit more about what the biggest challenges were. When you show those diagrams of model structure and hypotheses in your role-based modeling, they seem very straightforward, but I know building them is not. Well, if you think about the intellectual process of going from that image on the right, the toy model, to where you are now, what were the biggest challenges you faced in terms of understanding the biology, in terms of building the model, in terms of representation? What were the biggest challenges?
Yeah, that's a great question. Definitely for sure, they're really ironing out my agent-based modeling rules and like these cell dynamics, because there were things that I thought were very clear cut when I first defined my rules. And it wasn't until I was going through the calibration that I realized that like there were nuances that I like missed in my rules that just weren't accounted for, but like biologically there was something there that was preventing certain like things from happening in the model. And then also kind of thinking about like how much do I need in my model to answer my research question, because I feel like in the beginning I had this tendency to want to include everything, and had to figure out what to scale back and what was essential to model the things that I wanted to include, as well as just finding calibration data that is like comparing apples to apples. There's a lot of data available, but that doesn't necessarily mean that it's the right data to use, and then figuring out the most appropriate use of like setting criteria when I was doing my calibration so that I didn't narrow in my parameter space too quickly. I definitely had a bunch of iterations where I had to redefine criteria, and even just picking like how I wanted to set the criteria, because I kind of had to step back and think like, what is the most important thing for calibrating my model? And that was cross-sectional area recovery, because that's like the biggest model prediction. And then what cell types are most important for that, and then including satellite stem cell and fibroblast count, because those cells are pretty intertwined, and trying to calibrate one without the other would be problematic. So yeah, that was a lot, but those were definitely the big things that come to mind that were challenges as I developed the model.
Maybe we want to drill down a little bit more with that, because I really enjoy your tables where you have essentially what I'd call semi-quantitative data. You increase X, Y increases or decrease. Your tables with the squares with the upper and lower triangles to see whether the sign of the change was the same or not in the different perturbation experiments. And so those what I call semi-quantitative criteria, I thought, yes, this one was very nice, and it might be worth drilling down a little bit more. And now you're talking about also quantitative criteria. Is the amount right? Is the time right? And I wondered if you could talk, I mean, I know there's no universal, but about the balance between these semi-quantitative, qualitative, and potentially quantitative criteria for model constraint and evaluation.
Yeah, especially so when looking at experimental results, a lot of experimentalists, when they publish their paper, aren't necessarily thinking like, how can I format my data best for use of like model implementation? So oftentimes like the experimental results aren't necessarily quantitative enough to directly calibrate a model or compare like data for all these different time points, because often they'll just say like, oh, CSA was significantly higher with XYZ condition. So that's kind of where this type of comparison was most useful, because we were able to see how our like model was aligning with general consensus from these experimental studies. But the other piece that we had to kind of look at was at what time points that we would compare model simulation to experimental simulations. And so that was something that we had to kind of comb through. So we would look at all the experimental data and like their findings for each, and then look at our model and kind of compare across the time to see if there were inconsistencies, and then kind of what may be causing that, what conditions were in the experiment that maybe our model isn't capturing. So it also gave us kind of like next directions for where our model could be missing certain dynamics.
I mean, one thing, there was a talk in the Paris meeting 2 weeks ago by a group in Norway, and they were looking not as such complicated multiscale models, but more network models. And they start out by saying there's this tension between parameters, which are relatively easy to change, and model structure, which is a little bit harder to change. And so they were building a sort of a calipro-like system where you gave it a network, and it would change parameter values, rate constants, and all for the network. But it also would actually change the model structurally. It would say, what if this regulatory link were not there? Or what if there were a regulatory link that we didn't know about? And so it would actually generate clouds of models with different structures around the base model. And then use this kind of approach, to the calipro-like approach, where you have a bunch of scoring criteria. You get some fraction of those that are satisfied, and you have some threshold for accepting or not accepting the model. I know for CompuCell, automating, adding, and deleting mechanisms might not be so simple. You could probably boilerplate them all in with zero strengths, and then turn them on as needed. Would be possible. But I wondered if you imagined, for example, perturbing around the rule set that you have. Now here you have a fairly long list of criteria, but still not not anywhere near as many criteria as you have parameters. And so I wondered if you maybe could, how easy is it to find a model that will satisfy most of these criteria? There's the old saying, if you have enough parameters, you can fit an elephant. In my experience, that's not true modeling. It's very hard to get a model that will actually fit even a limited number of criteria. But I wondered if you could talk about that a little bit. In other words, do you think this is enough criteria to be fairly selective? Do you need a lot more? Do you need different kinds of criteria? What kinds of data sets maybe are missing that you'd need?
Yeah, I mean, for sure. So like here I tried to kind of capture a little bit of everything, right? So if we're knocking out different cells, like those dynamics, we wanted to see if we could capture that, as well as for removing cytokines, can we capture that? And then if we start like injecting, so like both can we enhance regeneration as well as decrease regeneration outcomes to make sure our model could capture both. But of course, there are more conditions that we could try to simulate. Specifically, if there were more experimental conditions in terms of like injecting different cytokine combinations, those would be really interesting to look at. The other aspect that you brought up about like kind of perturbing the model through calibration too. So that's another thing that we're interested in looking at. So like setting different variables to kind of turn on and off certain things. So like having a variable that will, turn off certain decays or diffusion, or will turn off cell recruitment, and then kind of simulating those to see how it's capturing, different types of conditions. And then we can also look at like adding in new unknown parameters in terms of looking at different treatments. So like if we introduce an unknown parameter that's like a concentration of a certain cytokine or the timing of delivery of something, we can then have that as an unknown, and then just set our calibration criteria to be that we want a certain amount of cross-sectional area recovery at a certain time point. And then that's kind of like a way to try and optimize treatments by introducing these new unknown parameters that correspond with like a specific understanding for treatment optimization.
Thank you again. Any final questions for our speaker today? Okay, well, I want to thank you again for a great talk and for your willingness to engage on these problems, and look forward to seeing how this progresses. Really excited by it, and we'll see everybody in 2 weeks. Thank you, everyone. Thank you again for coming.